Alpha-ketoglutarate plays a pivotal role in balancing carbon and nitrogen metabolism, and it is widely used in the food, pharmaceutical, and animal feed industries. However, microbial engineering for alpha-ketoglutarate production has long been limited by the inherent inefficiency of the metabolic network. A group of Chinese researchers, in their study, titled, Cell Factories and Transcription Factor Engineering for Bioproducts, the case of Candida glabrata for alpha-ketoglutarate production, set out to address this challenge. Transcription factors are known to regulate specific gene sets by binding to DNA and interacting with proteins. Building on this understanding, the team used transcription factor engineering to develop efficient microbial cell factories for alpha-ketoglutarate production. They reintroduced 3 transcription factors, GCR2 to activate the glycolytic pathway, RTG1 to boost the TCA cycle, and HAP4 to enhance the electron transport chain. These were then optimized to redistribute carbon flux and reduce overflow metabolism. As a result, the engineered strain, Candida glabrata KGA17, was capable of producing 210.4 grams per liter of alpha-ketoglutarate in a 5-liter bioreactor. This is the highest alpha-ketoglutarate production reported so far through microbial fermentation. These findings show that transcription factor engineering can create new opportunities for designing efficient microbial cell factories and advancing the biomanufacturing of chemicals. This paper appears in the December 2025 issue of Biofuel Research Journal and can be accessed for free at www.biofueljournal.com.